And we have today Dr. Eric Milbrandt. Eric is the director of the Marine Laboratory at the Sanibel Captiva Conservation Foundation. His research has focused on coastal ecosystems, specifically the role of natural and human disturbances on mangroves, oyster reefs, and macrophytes. He has his bachelor's in biology from Humboldt State University in California and his PhD in biology from the University of Oregon. So Eric, thank you so much for joining us virtually today. We're pleased to have you. Go ahead, you may share your screen and the floor is yours.
Yeah, great. Thank you so much for the invitation. And the talk today is about the Gulf of Mexico, but a lot of my work is in restoration, and I'd love to keep the conversation going after this seminar with some of the faculty. I know I have individual connections with various faculty members, but let's see.
Perfect. We can see it perfectly.
Okay, very good. Thank you. Yeah, as David mentioned, I work for a nonprofit on Sanibel Island. We've been an institution since 1967, primarily as a land trust to purchase undeveloped wetlands in the middle of our barrier island and save it forever and protect it and use different management strategies to promote wildlife use of that habitat. In the 1990s, the foundation recognized the need to establish a scientific presence in the marine science and oceanography. So as part of that, we started the Marine Laboratory, which I'm the director of, and we embarked on a variety of research, but mostly focused on our geographic area. So today I'm going to tell you about a large hypoxic event in the eastern Gulf of Mexico, and also several harmful algal blooms that occurred in 2018. And I know that this is a bit of a retrospective and descriptive talk, but I hope what we learned from this event is to increase our research and monitoring so that the damage may not be as great in the future if it's recognized more quickly. So I have a few co-authors, Natalie Nelson, former UF graduate and now assistant professor at NC State University, and Dr. Ed Flips, who is one of your colleagues at University of Florida. So dissolved oxygen, of course, is important for sustaining all marine life. It is produced, most of it probably in the global ocean, by microscopic cyanobacteria. And those were only really discovered in the 1980s. When you look at a map globally, of course, you see the sea surface oxygen concentrations in the tropics being lower than in the poles, and that's because temperature, higher temperatures can hold less oxygen in the water. Hypoxia is a term used to describe when the dissolved oxygen concentration is depleted to concentrations that are below typically 2 to 3 milligrams per liter. And this has severe consequences to marine life. The plots on the bottom of this slide show data from a review paper by Vacour, Saunier, and Duarte. And it shows on the left the sublethal thresholds for a variety of taxa. The fishes are probably the most susceptible to low dissolved oxygen events. But as you move down, you can see concentrations that are even at 5, 4, or 3 milligrams per liter of oxygen can be quite harmful to crustaceans, mollusks, cnidarians, and so we are recognizing that even below, not optimal, but even at middle dissolved oxygen concentrations, a typical well-mixed water column in the Gulf of Mexico will have 6 to 7 milligrams per liter for reference. Then the other thing that was recognized by these authors, which I think is important, is the lethal period, or the time that it takes when you're exposed to low oxygen conditions, and that there's a variety of responses by different organisms to that. But certainly the exposure that we saw in the Gulf of Mexico during 2018 far exceeded some of these times. So hypoxia in the world's oceans, interestingly enough, occurs naturally where the deep ocean is upwelled, and these are also some of the most productive fisheries in the world. Areas off of Chile, and as you can see off of the Mexico, you have intense upwelling followed by phytoplankton bloom. And not only can the deep water be hypoxic, but also the phytoplankton blooms will, as they disintegrate and degrade, that they'll use oxygen very rapidly and create hypoxic zones. Hypoxia can also be attributable to increased nutrients and runoff to estuaries and coastal areas. And as you can see on the graph on the right, this process of upwelling, but this is the eutrophication part of the story in the Gulf of Mexico, which everyone probably knows quite well. Hypoxia in the Gulf, a large zone, is driven by the load of nutrients from the Mississippi. In fact, it's now predictable because of intensive modeling efforts, and also the known flow volume from the Mississippi. Those combined are developed into a predictive area for this large hypoxic zone south of Louisiana. This area is about 15,000 square kilometers. And what's really, I think, groundbreaking about this is back in the 1990s, there wasn't such a thing as continuous observations in global oceans or near the coast. Most of our observations that were continuous were on the atmosphere. So Nancy Ravelet and others established a lot of these continuous observations. And since then, you can see the time series at the bottom there of the area, so of hypoxia since the 1980s to today. And there are policies related to this to try and decrease the size, or at least keep the area of that dead zone from expanding each year. And of course, you know, the warmer ocean temperatures and potentially changing climate conditions, such as all of the large major hurricanes that are affecting Louisiana now, have the potential to make these ecosystems even more vulnerable to hypoxic conditions. So based on this coast in Southwest Florida, I'm talking really from Tampa to Collier County or Naples, we do have commonly, almost every year, a bloom of algae that's by a dinoflagellate called Karenia brevis. Karenia is microscopic. You can see it's about 60 microns, and it can bloom to very high concentrations in the fall, typically. In the past 56 out of 62 years monitored, these blooms have occurred, and there are reports of blooms occurring in the 19th century as well. Karenia brevis produces brevetoxin, which is a neurotoxin that really has a huge effect on fish, in that it suspends their ability to move their gills and breathe, and so you end up with fish kills, which is one of the early warning detectors that is used is a fish kill hotline by the state of Florida and the Florida Fish and Wildlife Commission. Most of the impacts, as I mentioned, occur from Tampa Bay to Sanibel, but are actually quite limited in reality because of the patchiness of the blooms. The satellite image on the right shows the Sanibel coastline, Charlotte Harbor in 2019. So this is the year after the hypoxic event, but just to show you what a typical Florida red tide would look like from space. And you can see how patchy it is. So you may be at a beach on the west side of Sanibel and not feel any of the impacts or see any dead fish. One of the other impacts to people that happens with red tide is respiratory irritation, which is typically felt within a mile of a bloom. Well, up till 2018, hypoxic events, even though the concentrations of the cells were millions or tens of millions per liter, the decomposition did not seem to fuel any kind of further damage, like using all the oxygen in the water column. And that might be because of the patchy nature of Karenia. However, there's one exception. There's a report in the back of a paper that Weisberg published talking about a benthic die-off in 2005, which was a particularly powerful Karenia brevis bloom, but it wasn't really documented in the water column, other than some benthic species, like things that are usually in the sediment that will come to the surface if there's no oxygen, like punctulids and mollusks and other burrowing organisms. So not to revisit a nightmare, but 2018 was a terrible year for Florida algal blooms, and it may have had a very tremendous effect on voting that year, as we did vote in a new governor. But as you recall, 2017, September, most of us were evacuated from our homes in Southwest Florida and in the Keys because of Hurricane Irma. And of course, hurricanes are part of this ecosystem. They've been happening for a very long time. But a major landfall of a hurricane brings some tremendous changes and disturbance to not only the landscape, but also the amount of rainfall and runoff into the coastal estuaries and from the Lake Okeechobee, which we are connected to. So the red tide bloom from 2017 started in November. And we were able to, it was pretty limited to around Sanibel in November. And we were able to deploy an autonomous vehicle and sampled it. And then it kind of seemed to go away for a few months during the winter. However, it was enduring out there in the Gulf, and to come rear its ugly head again in August. So earlier in 2018, however, in May, the lake levels were very high, and the Army Corps of Engineers concerned about the dike around the lake being compromised and potentially flooding the communities around the lake, and decided to discharge water to lower lake levels in June, which is the hottest and driest month of the year here in Southwest Florida. And they had some information about the cyanobacteria bloom that was occurring from satellite imagery and from NOAA, which is a federal agency. They had some information that there was a large cyanobacterial bloom that had affected the east coast of Florida the previous year that was present. But the need to discharge the water was important, despite the fact that throughout the dry period in 2017, we were actually asking for water to maintain a balance of salinity in the Caloosahatchee River to protect some of the seagrass habitat that was there. And they released the water, and it created a massive problem of grand scale and affected everyone. The soupy green water had a major impact on the estuary. Cyanobacteria are freshwater species. Microcystis, the species that caused this bloom, it has a very good ability, not necessarily to grow and thrive, but it can survive, if you will, and still release toxins into the estuary, even at higher salinities, up to 20 parts per thousand. So while we don't really understand the interaction between the cyanobacteria freshwater bloom and the red tide bloom that occurred in the months after, there's a chain of events here that seem to be linked. So this is a beautiful shot of red tide off of Sanibel, the south end of Sanibel, looking east. It was shot by a gentleman who is a freelance photographer, and he went up on a parachute with a propeller on his back and got a really cool image. The hypoxic area really, it wasn't being sampled. I mean, the Gulf just in general wasn't being sampled, and our organization operates a wave buoy in that same area where I showed you the photograph south of Sanibel. And it was deployed the year before, but we went out to do some service on it, and I had to send divers down to attach the mooring, and discovered when we went out there a very strong sulfide smell. Went down, there was a black sulfide layer on the surface of the sediment that no one had ever seen as in previous dives. And of course, when I went down and picked up it was a fighting conch, the inside of it like melted in my hand in the water. It was really pretty gross. But at that point, we were like, well, there's something going on here that's we need to document. So, and at the same time, I should say, this is August of 2018. There were some cruises going on to sample red tide, and there are reports from my colleagues at FWRI and at NOAA who sampled during the time that we were making these observations. So at any rate, we established kind of an event response. We have 3 permanent continuous monitoring stations near the mouth of the Caloosahatchee estuary. Those are indicated by the plus symbols. Those have been maintained and are continuous since 2008, and have recorded dissolved oxygen, salinity, chlorophyll, turbidity, a variety of factors related to water quality. And the circles are the grid type sampling that was adapted from the NOAA sampling effort in the Northern Gulf to describe the Northern Gulf of Mexico hypoxic zone, which is a series of transects offshore up to 10 miles that we could do with our small vessels. We have 2 23-foot vessels, so we couldn't go all the way out where we really could have defined the edge, as it turns out, but I'll tell you more about that story in a second. We also initiated a series of sampling sites related to the interaction between the estuary and the nearshore Gulf of Mexico and the discharge sources through an NSF Rapid grant. And those sites are indicated in triangles. We actually had sites all the way up the Caloosahatchee to sort of understand the fate and the phytoplankton communities along that estuarine gradient. But I'm only going to really be talking about sites in the Gulf of Mexico today, because this talk is mostly about the hypoxic event. So when we went out to deploy our profiling CTD, we used a calibrated YSI XO2 SON set to record every second. And this represents 3 different sites. Each site is graphed the chlorophyll A, which should probably just be relative fluorescence. That is a fluorometric measurement. And then salinity is in red, temperature is in blue, and dissolved oxygen is in black. And on all of these graphs, I want you to first just look at the black line, the dissolved oxygen. It's fairly high, some cases very high, above 8, 10 to 12 at the surface. And then it declines as you get deeper to almost 0 in some cases, and in other cases down to about 3. Now, I guess if you're not used to looking at this type of graph of the water column, the depth is the y-axis. So as you get deeper, if you look at salinity, it increases. So it's fresher water at the surface and saltier water at depth. And then temperature, it's higher at the surface and colder at depth. So it's warmer at the surface. So what this is telling us is that the warmer, less salty water was separated from a layer of water in the bottom of the water column. And that layer was fairly thick and would not mix with the surface water. So there was a density difference that prevented any mixing from happening. So this is more just descriptive. You can see at all of the sites that we sampled a layer of low oxygen water. That would be the darker blue as opposed to the lighter blue. And a percent, we calculated a percent hypoxia within each of those samples, and it was around 50%, but it varied depending on the depth. Another way to look at the dissolved oxygen is to look at the depth profile, the bathymetry together. And the purple is the very low oxygen or hypoxic water. Again, it's this isolated layer within a water column that was well stratified, which is quite unusual for this part of the Gulf of Mexico. And if you use data interpolation and variational analysis, it's just a way of interpreting between those CTD casts. You can kind of see that layer more visually. And also in the lower graph, the more red section is the saltier water, and the lighter yellow and green colors is a little bit more fresh. We're not talking like fresh water like an estuary, but there was a big enough difference between the saltier bottom water and the fresher water to create this stratification. We think what's going on in the DO in the deeper water, that's not an upwelling event or new water from farther out in deeper water. What that is the sample was collected over a large hard bottom reef area that we've surveyed many times, and it has abundant macroalgae. So we think actually there was photosynthesis and production of oxygen near there. So we used a tool that is developed off the shelf within ArcGIS to interpolate the different layers of water. And just to orient you to what this series of graphs is, it's a series of slices through a modeled water column that has, the model is the dissolved oxygen concentration. So the red is hypoxic water, the blue is normoxic water, and then there's also some really high oxygenated water, supersaturated water. also as a category. And supersaturated would be greater than 8 milligrams per liter. Hypoxic was less than 3 milligrams per liter, and normoxic, everything else. So as you go through the slices, as you start at the surface and go down a meter at a time, you can see again kind of how the layer was around 4 meters, between 4 and 5 meters deep at all of the sites, and was set up by this density difference. So one thing that we didn't have is a lot of continuous measurements, but we do have them near the estuary. And one of the things that we noticed is that the dissolved oxygen, the hypoxic water, was actually moving past the sensor at certain times. If you look at the dates there, around the 15th of September and again around the 29th of September, these are periods where the tidal regime went from more of a neap tide to a spring tide. And during those times at a flood tide, the low DO water was moving past the sensor. And if you plot up depth versus DO, as I did on the lower two graphs, during these spring tide periods, you can see a significant negative correlation between DO and depth. So what that's telling us is that water, as the tide comes in, the water with low dissolved oxygen is coming in. The source of that water is the Gulf of Mexico and not the estuary, which is kind of unusual. So over about a year period, we continued to sample at a subset of sites in the Gulf. This plot just shows the difference between surface and bottom. The surface is black and the bottom is gray. The dissolved oxygen concentration in September of 2018, and then continuing through the next year at various sampling events, all the way through November of 2019. I show in the middle graph a sigma T, which is a density calculation based on salinity and temperature. And you can see that density difference that I was describing. And it was more the salinity difference than the temperature difference in our analysis that was driving that density difference. In the bottom graph is the Karenia dinoflagellate concentrations, and this is the units are in grams carbon rather than cells per liter. But you can see most of the cells are concentrated at the surface because Karenia is a dinoflagellate that can swim typically towards the light during the day and can swim down to get nutrients at night. And there's been some great research done by Cindy Heil and others that show that these layers near the surface actually have their own biogeochemistry that's totally different than the rest of the water column. But you also notice that in 2019, there wasn't any hypoxia or difference in dissolved oxygen, but there was still a significantly intense red tide boom. So to re-emphasize the effects of Karenia in this situation with dissolved oxygen, the places where there was really sort of remarkable numbers of dissolved oxygen were on the southern part of Sanibel, where one sample was counted with 47 million cells per liter, which is, most of the time, if you are familiar with the Karenia and counts around the state, they target certain beaches and they are sampled routinely, weekly usually. And a lot of times the concentrations may or may not reflect the actual concentrations in the Gulf of Mexico because they're being concentrated there. I mean, beaches they concentrate things, whether it's macroalgae drifting past, whether it's organisms that are washed ashore or Karenia. And so we were quite surprised to see really high concentrations like this in some of the patches that were offshore. And it created supersaturated areas in the water column. So associated with this, all of these things that were happening, there was a mass mortality event. The Sanibel had to, and much of Lee County, decided, even though that we don't rake beaches, we want the beach to be as natural as possible on Sanibel. It was such a big event and overwhelming that a contractor was used to remove over 370,000 kilograms of dead marine life from the beaches, including 82 Goliath groupers, which some of them were very unusual in size and hard to sample. So the FWC fisheries folks were here getting otoliths and so on. 21 tarpon, and there was over $1 million spent on that process of removing dead stuff. There was also statewide manatee mass mortality. So one of the things that in our event response we decided to try and do is to document some of, I'm a biologist, so I'm much more interested in what's happening in the benthic areas and how it affects life rather than the DO measurements, but obviously those are the things that are causing a lot of this. So we decided to just do a qualitative analysis, and we went to several areas and documented what was there with video. And since everyone loves to see videos, I'm just going to show you a very quick, it's not the greatest color because of the filter set that we had on at the time, but this is the recording by a diver during the hypoxic event. And it was on an artificial reef, so you see some culverts there. There's a herring. And you can see that the color of the sediment was, it was pretty dark. There were several large black patches. All right, so we returned to some of these artificial reefs later, and here's the result. You can already see several spadefish there, and just the difference in terms of fish. Obviously, they were, most of them had during the hypoxic event had been killed likely, and a lot of snapper. A lot of anything with structure in the Gulf will attract fish. Hey, my videos worked. Well, just to continue on the timeline, later that year, one of the actually most destructive hurricanes for our colleagues at the Florida State University Marine Lab was Hurricane Michael, and did a lot of damage to other places too. But it went past Sanibel in October 2018. There were about 40 hours of wave heights over a meter. And we were able to record that with our wave buoy that really started our journey down this path of trying to document this event. And there was even 19 hours of wave heights over 2 meters. So after that significant mixing event, there was no longer low DO levels, and we have not seen those levels of hypoxia since then, including up to today. So just to summarize, if I haven't gotten the point across already, that stratification was likely the main reason why the water column had become hypoxic and isolated from mixing with air and oxygen. This event occurred for about 8 weeks, so far as we can tell. So there's been some suggestions and discussion, although we'll never be able to probably demonstrate it, but that fresher, warmer water on the surface was from record high flows after Hurricane Irma through the Caloosahatchee and the Peace River, Myakka River after Irma. There was 100 days of flows greater than 10,000 cubic feet per second, which is about 3 times the harm threshold for the Caloosahatchee estuary. The mass mortality event, I think it may have been at least initiated by some toxicosis of fish from Karenia. But I think the 600 should be a square kilometer, not cubic kilometer, sorry. 600 square kilometer hypoxic area that we could measure, which we never found the edge. So that's probably a conservative estimate. Likely had a more profound effect on the amount of dead marine life on the beaches and elsewhere. We even had a whale shark wash up, which is pretty incredible. So the year following, in 2019, we had a pretty strong red tide with high concentrations of Karenia, but there was no signs of hypoxia. So I guess my take-home message, and maybe this is a message for all of the scientists that are out there, that are maybe the students especially, we need to sort of really be critical about what kinds of sampling and monitoring that we're doing, and make sure that the limited amount of dollars that's available for that is being spent in the best way that we can. Because this event-driven sampling is not adequate to really help us predict or determine characteristics of what happened and why. We need modeling, of course, which is actually very good in the state of Florida, but we actually need people sampling and collecting data, and that's an important part of being able to run the models and predict processes that are happening. So more dissolved oxygen monitoring is needed in the Eastern Gulf. If you look at the map below, that was a screen capture that I took yesterday. The FWC does a great job. They have to cover the whole state for red tide. And you can see that the sampling is not, there's no sampling in the last 2 weeks being conducted offshore. I know they're trying to do more. There's funding for mitigation of red tide and early warning and so on. And yet there's just a serious lack of sample collection in this zone. And they are, the Florida DEP in their regulatory reach does have zones that are in the Gulf of Mexico for looking at different impaired waterways. So I think at least we need to be sampling near shore for those. So we're doing our best to chase proposals and funds to try and do it. But I think we really need to understand this area more in order to help us with climate change and sea level rise and warming of ocean temperatures. It's become apparent that HABs aren't going away, and they really need to be understood to try and decrease the frequency and intensity of them. And I think I'll leave you all with a shot of a rainbow and some really some funding acknowledgements there, and also my staff that have gone out on various occasions and sampled during the event.
Great, thanks so much, Eric. So do we have any questions for Eric from either from YouTube out there or from folks in the Zoom room? I'll start you off with one, and maybe the answer is it's complicated or you don't know, but you noted that it was, you didn't necessarily have the capacity to differentiate, hypoxia induced mortality versus, Karenia brevotoxin mortality, and like not a physiologist at all, but is that something like, some of these fish necropsies can do for you, or is it just that they're so interwoven that it's hard to disentangle?
Yeah, there are definitely some tissue tests that were being run during the time. For example, we are the permittees for sea turtles, and there was record stranding that year. We did sample a lot of those dead turtles and their guts, and there were loaded. A lot of times the guts were loaded with filter feeders, pen shells, and so on. And so the hypoxia for a turtle is not really a problem, right? Because they can surface and breathe. But what they were eating in their diet was totally toxic and killing them. As far as the fish goes, I think it's probably not possible unless you were had a continuous type data. You could track a population through an event, and a good fish biologist, I'm sure, could come up with a sampling design.
We just need sensors for DO on all the fish, right, as they swim through their lives, and we can get the data directly.
They may not be that far off, but we can put acoustic tags and figure out where they are.
Yeah.
The technology is really moving quickly.
Andrew, I see you, and you have a question?
Yeah, Eric, that was a nice talk, thanks. I actually saw some of that material at SURF. I think one of your staff members may have presented there. So it's nice to kind of see some of this additional interpretation you provided and some more context in this format. So thanks for that presentation. I was just curious, so you noted at the end of your talk that 2019, there was a red tide event that was not associated with hypoxia, at least detected. I'm just curious if you have any ideas of why that might be and what clues that might offer as to how hypoxia is, the origins of the hypoxia in your area.
Well, I think there's two things. The 2019 event was more patchy, and it moved around. The patches moved. The 2018 event was like, it was everywhere, and it seemed like it was stationary. I know it wasn't. The ocean is constantly moving and dynamic, and there's currents working on it all the time. But it seemed to sit there for a long time. And to me, if you had that kind of production as the bloom declined, it would certainly increase the hypoxia, the amount of oxygen demand that was in that lower water. But I think the key is the discharges from the rivers after the hurricane likely set up a plume, if you want to think of it that way, but it was an area of fresher water that was either resistant to mixing during that summer, as opposed to the 2019 year. That's kind of about it, as much as I can say. But what I really I've had some conversations with the modelers that can they really have a good understanding of some of the drivers in the deeper ocean for red tide. There's particular things that they look for in the loop current and so on. But this, to me, because it was a nearshore event in shallow water, and talking to them, seems like most likely to be from runoff.
Right. Okay. And so you're tying though the low oxygen to the red tide itself? I mean, caused by the red tide, or just that they have a common cause?
So I think I think they have a common cause, and that it was made that the hypoxia was first, there's some feedbacks going on. It was made worse by the input of toxicosis fish. And then I think the red tide wasn't really killing everything. It was the hypoxia at some point.
Yeah.
And after some period of time, 24 hours, 48 hours, that's when we saw this mass mortality event.
Mm-hmm.
But it would be kind of interesting to not only measure the oxygen demand of the sediments, because a colleague of mine, who you all probably know, Dave Tomasco, measured hypoxic demand in Charlotte Harbor sediments that led to the stratification was from fresh water, but the oxygen demand from the sediments in Charlotte Harbor within a few days would completely deplete the lower layer from oxygen. And so I think there can be something learned from, if you just isolate the water column, how long does it take to go hypoxic? That's something I think that we should know, and maybe we can model it, but we could also measure it.
Yeah, and sorry, I don't want to dominate things, but I just have one follow-up question. So I'm just curious if they're measuring concentrations of the Carina brevis at depth, and whether the depletion of low oxygen closer to the bottom is either because, I mean, if you can kind of surmise whether it's because of the actual respiratory demand of those algae, at night, or whether it's because they're dying and sinking to the bottom, and it's the decomposition of their biomass, or if you have a sense for kind of like, just those processes at all based on your data.
Yeah, I'm not as familiar with the Carina literature as probably other people, but I'm sure that someone has taken 1 million cells per liter and set it in the lab and looked at oxygen demand. It could probably do some calculations based on those measurements. But a lot of times our Carina concentrations are more reactive, and the sites where they're collected is reactive to a bloom. And so I really think we need systematic grid type sampling that samples the surface and the bottom, just to get the basic information. And then I think you brought up a number of really interesting processes that should be studied further.
Yeah, thank you.
Christine, you have a question?
Sure. I just want to make sure there wasn't any from the chat or anything. I don't want to have absorb the.
So far, so good. I'm pumping YouTube folks, but I'm not getting anything. So.
All right. This was very interesting, Eric. I'm kind of fascinated that, essentially you have one hurricane setting up the situation. And then from what you talked about, it took the second hurricane so one set up the stratification, and then it took a second hurricane to basically knock it back to like a more mixed state, which is pretty fascinating in terms of basically the stability of these types of features. But it seems to me that basically when you have these freshwater blooms, that it drives a big lens offshore, that it's fresher, it's, and you have this enhanced productivity in the upper sort of surface layers. And it really is that trapping of the salt water kind of underneath that sets up the potential sort of bottom water hypoxia to sort of happen. And so I'm wondering on the modeling side, from what I understand of what Weisberg does, it's very much from the ocean sort of pushing stuff on shore. But what you're talking about is really fundamentally understanding the riverine coastal to ocean sort of like process and the potential for ocean water to trap that sort of freshwater lens at the coast. And so I'm wondering what the next steps in terms of modeling are, and are they really building out from like the Caloosahatchee and the Myakka River and looking at the interaction between that sort of that head from the river and like the ocean, sort of what would be sort of pushed on shore by the eddies and the sort of the feedbacks between those processes, right?
Yeah, so I'm obviously not a modeler at all, but I think what I've read in the Carina literature and the modeling literature is people think that the Gulf of Mexico and the West Florida Shelf has such a high volume that you could never really modify it, except maybe in a few localized areas to cause something like this. But I think really as we get more extreme rainfall events and 100-year floods every few years, that any connection between the estuary models and the Gulf, I know they do connect.
Yeah.
Based on what I've seen, there's an ocean, usually an ocean end member and the river end member, but I think in particular with the Caloosahatchee, that area south of Sanibel has historically been one of the main Karenia sites. And I know recently one of your colleagues published a paper that correlates freshwater and total nitrogen to Karenia along the coast. So maybe that's a sort of a first step to get to that. That, I mean, it's a physical occurrence, so it has to be predictable, right?
Yeah. Anyways, thank you. It was educational, and I think it's most likely you're dead on that this multiple stressors thing, it's not just Karenia, but the double hit of the low oxygen events kind of causing mortalities seems really I don't know. I learned that today, so thank you for that. Yeah.
Yeah, thanks so much, Eric. So everyone either, unmute and clap or clap, 21st century clap with your little emoticons here. We thank you very much for joining us today and appreciate the talk. I'll clap for real. And hadn't had a chance to meet you before, so nice to meet you. And we'll be keeping in touch. I know you connected with Christine and others, and we're very keen on trying to couple this sort of onshore and offshore processes. So I'm sure we'll be talking more.
Yeah, great. Thanks again for the invitation.
It was a pleasure. And of course, students, turn in your one-page summary. Dr. Clark and I will get to them, I'm sure, at some point. And until then, we will see you next week. Thanks, folks.
Thanks,
Cheers.
Eric. Nice
Bye.
talk.